Nataqain binti Baharum, your moderator for today's session. I hope the video has provided you with some background. So for those who are interested to collaborate with us, you are most welcome to do so. Please feel free to browse INBIOSIS website and social media for the latest updates and info. I feel so grateful today that all of us are granted good health and time to attend our first webinar collaboration with AHS Chula. So I would like to elaborate a bit on the Allied Health Science Chulalongkorn University. It was founded in 1992 as an advanced academic center for health services. The faculty prepares its graduates with the knowledge, skills required to professionally address illness in holistic manner, meeting international standards and ethics. The faculty produce graduates with professional expertise in 4 main areas: physical therapy, nutrition and dietetic, radiological technology, and medical technology. It is also a pioneer in conducting quality research and developing innovations in advanced health science for the benefits of the society. There will be many more webinars to come, and I hope all of you will join again in the future. INBIOSIS webinar series is established to provide a knowledge and research experience sharing platform to ensure we are still connected with each other and stay motivated in our research despite these sadness pandemics. I'll now give some housekeeping rules for our webinar today. The lecture will be taking about 45 minutes and 15 minutes for the Q&A sessions. And maybe we'll take a bit longer depending on the numbers of questions posted. Please submit the question in the Cisco WebEx chat box and FB Live page. I will read all the questions during the Q&A session. Before the seminars end, I would like to invite all the participants to switch on your camera for a photography session. Please stay tuned until the end of the session to fill in the attendance form for e-certificate and Urus Bakat UKM credit. Without further ado, I would like to introduce our honorable speaker for today, Dr. Tewin Tencomnao from Chulalongkorn University, Thailand. Allow me to share Dr. Tewin's CV. Dr. Tewin has focused his research on the natural product for the neurodegeneration and anti-aging at Chulalongkorn University. With more than 4,500 citations and H-index 19, he has successfully upgraded the research quality in the field as evidenced by the publication more than 10 papers per year. His research team employ both neuronal cell culture model and animal model for the studies in order to elucidate the mechanism of natural product to fight against neurotoxicity, neuroinflammation, neurodegeneration, and aging. So let us welcome Dr. Tewin to share his research finding. So Dr. Tewin, the floor is yours. Thank you.
Thank you very much, Dr. Syarul, for the very kind introduction. So I'm really pleased to give a talk today. And actually, I'm really honored to have a chance to visit your university and also INBIOSIS and met the director 4 or 5 years back. And then I'm really happy to meet you all again today. And hopefully, after the COVID-19 outbreak, we can meet and collaborate again for our long-term collaboration. So thank you very much. So I just want to share with you my recent study on tiger mushroom. So let me share the slide. Can you see this slide?
Yes.
Okay, so I will start from the beginning. So thanks again for this invitation. So may I start now? So can you hear me clearly?
Yeah, loud and clear.
Yeah, so we have about 45 minutes for this talk and then QA session, like you mentioned. So I don't know how to point out using the laser or anything using the computer. So webinar also very new for me. We used Zoom before, okay? So this work actually done last year, and then we published 2-3 papers last year and this year. So you can see the name of the collaborators, mechanisms of tiger milk mushroom for anti-aging and anti-neurodegeneration. So this work done primarily by my student Parini Kiti Mungkon Suk, and then the collaborators, co-advisor Dr. Siriporn Chichwankun. And we also did this work partly in Germany, Heidelberg University, Professor Michael Wink. So I'll be the presenter today. So I would like to talk a bit the background significance, and then we go through the experiments and results and brief discussion and conclusion. You know, in Thailand, it's very serious in terms of aging nowadays. We have a lot of proportion people who older than 60 years old and also 65 years old. So this graph showing now is a period of 1 century, starting from 1950 to 2050. So in the period of 1 century, 100 years, we have the rapid increase of age people. As you know, now about 2021, right? We have 20%, and then it will be 30% in 2050. So about next 30 years. So this is very rapid increase for both 60 years old and also 65 years old, which we call super age. As compared to other countries in these Asia countries, in 2015, Japan is #1 in terms of aging, you know, and Thailand's in the top 10 of this list. For the next 15 years, 2035, Thailand will be in this list like #6, and Japan still #1, and Singapore #4. So you know, it's very rapid increase in terms of aging. We have low child growth, and we have a lot of aged people. So we have to do well prepare in terms of the future, particularly in my area of health improvement of people. It's okay that we want to have people to live longer than before, but the quality of life must be developed together. I mean, so in Thailand, so that's why the government policy will eventually apply to the universities and also on sectors, both government and private sectors, that we want to have people with good quality of life, no matter how long that they can live, actually. When we have a high proportion of people with high age, so we enter the aging society, and of course, that the age-associated disease are high risk in this group of people. Those diseases include cancer, stroke, type 2 diabetes, arthritis, heart disease, wrinkles, and frailty. For my area of research, I focus on neurodegenerative disease such as Parkinson's disease, Alzheimer. Based on this slide, as you can see, that the causes of aging derive from both exogenous production of reactive oxygen species, or ROS, as well as endogenous production of reactive oxygen species. You know, inflammation and environment are examples of exogenous production of reactive oxygen species, whereas mitochondria, cell respiration, cellular metabolism are examples of endogenous production of reactive oxygen species. So it's very hard to not to have the reactive oxygen species, because If we block one pathway, there will be some other pathways that leading to the generation of reactive oxygen species. So we consider reactive oxygen species as cause of aging. As we get older every day, every month, and every year, but it's very hard to to block the age, not older than yesterday. So can we think of other way that we can delay cause of aging? So this is the very big question of my research. Based on the theory, it's very complex process to detoxify reactive oxygen species, but the cells has very elegant manners, very well designed process to detoxify the reactive oxygen species based on step 1 and 2 and 3. Step 1, actually, whenever the superoxide anion oxygen radicals produced, the superoxide dismutase enzyme will convert the radicals to oxygens and hydrogen peroxides. Oxygen produced from this process is not toxic. It's good, but hydrogen peroxide that happen and occur in the cells. So the cells must have the way to get rid of the hydrogen peroxides. It could be step 1, or it could be step 2 or 3. Step 2, the 2 molecules of hydrogen peroxide can be converted to oxygen and water using the enzyme called catalase. Otherwise, it can be step 3, that hydrogen peroxide will be get rid of from the cell by glutathione mechanisms. So these 3 steps are very important, but we have to say that step 1 is the most important one. So if the step 1 doesn't happen properly, it will not go to step 2 and 3. And step 1, to get rid of or to detoxify the reactive oxygen species, is very crucial step. And based on the previous studies, there's some antioxidants from the plants, from the natural products, may work in this pathway as well. So back to my laboratory. So we use, actually there's so many models for this study. So particularly in the lab that I'm using now with the team in the group we call natural product group or anti-aging group at Chulalongkorn University. We use both in vitro models such as cell lines. There are a lot of cell lines that we can work on. For me, the neuronal cell lines are the focus because we want to work on in the field of neuroscience. And I want to show you today both in vitro and in vivo model. For in vivo model, we use C. ligands, the organism that have a short-lived style that we can count number of the worms based on this experiment, and we can also repeat so many times in the week, in the month. So this is very good model. If we use the big model like mouse, it will be so many months or a year that's very hard for studying the life expectancy of the animal. In the field of neuroscience that I work on, to increase the cellular reactive oxygen species, we use glutamate. From in the brain, there are glutamates that work as a neurotransmitter. So glutamate in the brain, there's a lot, but if we have only optimal amount of glutamate, low amount of glutamate, the brain can work so well. The cell has a way to metabolize glutamate, produce glutamate, and also destroy or convert glutamate to other forms in the brain. There would be no problem at all. However, if we have excessive amount of glutamate, it will lead to cell death because glutamate eventually can lead to the production of reactive oxygen species. So glutamate is very important molecule, and it can cause oxidative stress. So glutamate causing oxidative stress, causing reactive oxygen species. There are two pathways. One we call glutamate receptor dependent pathway. Second one we call glutamate receptor independent pathway. We call receptor independent pathway, meaning that some model of the cells, like the one that I want to tell you today, the cells don't have the receptor for glutamate. So since the cell don't have the receptor for glutamate, the glutamate accumulation actually can lead to the reactive oxygen species via the transporter mechanism. It doesn't have the receptor, so this cell line have no receptor for glutamate. So whenever the glutamate very high in the cell, in the environment of the brain cells, the glutamate will exchange with the cysteine. So glutamate will go inside the cell, the cysteine will go out from the cell. Whenever the cysteine go out from the cell, the glutathione will be dropped. Glutathione is a major antioxidant molecule in the cell. So whenever the glutathione drops, it will be not so good for the cell, because whenever the lower level glutathione or GSH happen, reactive oxygen species will increase. In that particular status, the cell will not be healthy. And whenever the cysteine go out, the glutamate go inside the cell, one molecule that is calcium will be in the status that we call influx of calcium. So when the cell have the high level of calcium, that will further increase the reactive oxygen species levels. Then the reactive oxygen species level will cause the damage of DNA. Whenever the cell have DNA damage, that will lead to apoptosis eventually. So the cell that we use, we call mouse hippocampal HT22 cells. So it was generated from the mouse hippocampal part of the brain. So we call for short HT22. The HT22 cells do not express receptor for glutamate. So whenever we use this cell type, we call non-receptor mediated oxidative glutamate toxicity. The cell don't express glutamate receptor, as I mentioned. Finally, reactive oxygen species occur will lead to the mitochondria oxidative stress and leading to cell death. Another model that I want to show you today, Rapid elegans or C. elegans, a free-living, transparent roundworm. They share high homology with mammalian and human genes and biochemical pathways. They eat bacteria as their food. The bacteria that proper for C. elegans we call E. coli of 50. So using elegans, one benefit that I can recall is, doing these experiments, it costs less as compared to cell culture. Cell culture, we have to spend a lot of money. We have to have a very good grant funding to do cell culture work. But for silicons, they are, animals, but not so expensive to work using silicons as a model. So one of the benefits that in the lab, we really prefer this model. And, silicons is animal. So study using animals in many mechanisms, we can get a proper answer. They are hermaphrodite, self-fertilization, average lifespan 2-3 weeks. So when we study the survival, when we study the anti-aging activity of the natural products, we can repeat experiment many times because 2-3 weeks, I mean, in one semester, you can do many experiments, many rounds of experiments. And silicons known for anti-aging, stress resistant, neuroscience, and longevity study. This slide showing the life cycle of the silicons. They are, they are lava stage 1, lava stage 2, lava stage 3 and 4, and then adult, and then after that they have gastrula, coma, and this is the normally the lifespan of the silicons, but 2-3 weeks only that they can live anyway. As you can see in this picture, the silicons model have 3 basic systems completed. Number 1, digestive system. They can eat, they eat bacteria. Number 2, reproductive system. That we can count the number of eggs of silicons. If the natural product that treat silicons are toxic to silicon, number of eggs the silicon can lay, normally 300 to 400 eggs, will be decreased. That means that the natural product cause infertile, so something like that. So we can interpret the data as well. And the third system work very well in silicons is the simple brain system, simple neuronal system. So 3 basic systems found in silicons. The natural product used for this study are tiger milk mushroom, Lignosus rhinoceros, or LR for short. Habitat mainly Malaysia and other Southeast Asian countries, including Thailand. Property in the previous study, the tiger milk mushroom can be used for treatment of asthma, anticoagulant, anti-inflammation, antimicrobial, antioxidant, anti-tumor, and promotion of neural growth in PC12 cells, and anti-HIV activity. So there are 3 parts, you see, in this picture of the mushroom: cap, stem, and sclerotia. Sclerotia or tuber is the part that we use for extract and then test in the study. So this part it's very important, sclerotium. The samples of the tiger milk mushroom was provided by the collaborator, Ligno Biotech, Malaysia. Anyway, little known about the neuroprotective effect of tiger milk mushroom extracts. So the hypothesis for our study, the tiger milk mushroom extract has neuroprotective effect against oxidative stress in HT22 cells via decrease in apoptosis and increased antioxidant gene expression. And the tiger milk mushroom extract exhibit both neuroprotective effect in cell line and also in silicons via the proposed signaling pathway such as DAF16 or FOXO, SKN1 or NRF2 signaling pathway, as well as decrease in aging marker. So I have to point out that using silicons, there's some marker in silicon when they get older, they will generate that pigment, which is autofluorescence. So we can quantify the autofluorescence in aged silicon easily. So that's one of the marker. And also other marker would be heat shock protein as well. So the objectives include investigation, the biological activity of tiger milk mushroom in HT22 cells and in silicons, and also determination of the mechanisms of neuroprotection and anti-aging involved. So the experimental flow shown in this picture. So first, the extraction that I want to tell you a bit. we have three different fractions. Number one, ethanol extraction, we call LRE, LR, the tiger milk mushroom, and E, ethanol fraction. Cold water extract, LRC, C, cold water, as well as the last one, hot water extraction, LRH, H for hot water. So we check antioxidant capacity testing using different methods like DPPH, ABTH radical scavenging assay, and we check total phenolic and total flavonoid content. And also we determine the profiles of phytochemicals in the extract using GCMS, LCMS, mass spectrometry. And next, we study in vitro model, and finally in vivo model. For the PPH and ABTS assays, scavenging activity found as in table 1, we found that the PPH assay, the results show the high activity of tiger milk mushroom ethanolic extract, higher than cold water and hot water extract. For ABTS assay, the table number 2, we also found the highest activity in tiger milk mushroom ethanolic extract, higher than hot and cold water extract. So it seems that for next experiments, we really focus on the ethanolic extract. So based on this initial study, we can narrow down which of the 3 extracts could be our focus. Next, we assay for the phenolic content and flavonoid content. So we found that for total phenolic, the tiger milk ethanolic extract has the highest level of phenolic content, whereas the flavonoid contents we found highest level not in the ethanolic extract but in the hot water extract. So this can be explained because the total phenolic and total flavonoid contents are different things, different properties. So it is possible for sure that different extracts, different type of solvent used would result to different contents of phenolic and flavonoids. So for the next experiment, we determine the phytochemicals contents using mass spec, and we found different compounds ranging from terpenen 4, phenol, glycopyranoside, oleic acids, and other phytochemicals, which actually previously our colleagues at Chulalongkorn University Faculty Allied Health Science, they determined the profiles phytochemicals in the aqueous extract. The possible phytochemical contents consist of high triglycerol, oleanic acid, and betulinic acids. So they published last year actually. So some of the compounds may be similar, some may be different, but again, it could be the difference in terms of the extract used for the study, and also different samples that we receive in the lab. Next, I would like to tell you more about the study in cell lines, which in the lab, if you want to check the number of cells, so the survival assay of the cell, viability of cell, we use MTT assay. So I will go to the result using MTT assay. And I want to show you, as you can see the red arrow. As I mentioned in the beginning, the glutamate GLU glutamate, 5 millimolar of glutamate can kill the cells. So based on the cell viability assay, as you can see, 5 millimolar of glutamate leading to cell death 50% roughly. So why the glutamate kill cells? I told you already that the glutamate will lead to the generation of reactive oxygen species. Reactive oxygen species cause the defective situation in nucleus, causing DNA fragmentation and leading to cell death. So that's why we can find the number of cells, survival cell only 50% after glutamate treatment, because glutamate can cause oxidative stress. So next we ask the question, which type of tigermeal mushroom extract? Hot water, cold water, or ethanol extract can work against the toxicity of glutamate? As you can see now, even the glutamate 5 millimolar, 5 millimolar of glutamate cannot kill the cells if the cells are treated with the tigermeal mushroom ethanolic extract. So you can see it works in the concentration dependent manner, meaning that higher level of ethanolic extract of tigermeal mushroom will have a higher protection. So less cell death will found in this experiment. But for cold and hot extract of tigermeal, the cells still die. So the protection that we found against glutamate toxicity is only found in the presence of tigermeal mushroom ethanolic extract only, not cold or hot water extract of tiger milk. Next, we want to check actually if the cell death due to glutamate is happened because of the apoptosis. To check apoptosis, we have to stain the cell with a Nexin 5 and propionium iodide or PI staining, and then check using the flow cytometer. As you can see, graph figure A here, that 5 millimolar of glutamate leading to apoptosis cell, about 60% apoptotic cell death. And then in the presence of tigermeal mushroom ethanolic extract, 100, 200 microgram per mil, the number of cell deaths based on apoptosis was reduced significantly. And we also compare with NAC, which is a positive control. Whereas tigermeal mushroom, cold extract, and water extract cannot prevent apoptotic cell death. And below panel of this graph, of this slide, you can see the flow cytometer result. And then we convert to the calculation to the graph. One mechanism leading to cell death happened at the organelle glutamate. As you can see, whenever the glutamate, whenever the mitochondria is healthy, the mitochondria, healthy mitochondria can stand very well with the dye we call TMRE, tetramethylrhodamine ethyl ester perchlorate. So this dye actually will stand in the intact mitochondria. So the high intensity of staining will be found in the healthy mitochondria, which is the intact mitochondria. Whenever the membrane depolarization happens, we see the dying mitochondria, which is unhealthy mitochondria. The level of staining with TMRE will be low. So this is the method actually for experimental approach. we grow the cell, we treated the cell with the extract, which is tiger milk mushroom extract, and we add CCP, and we also stain CCP, actually membrane depolarization. We stain TMRE, and we measure the fluorescence. As you can see, 5 glutamate, 5 millimolar glutamate, you can see the fluorescent intensity low. Because why it is low? Because the staining level will be decreased, because the membrane is not healthy. Mitochondrial membrane is not healthy, meaning that glutamate causes unhealthy mitochondrial membrane. So whenever we have glutamate, but we also have the extract of tiger milk mushroom, particularly ethanolic extract, we can see the high fluorescent intensity, meaning that whenever the extract still in the cell culture with treated cells, the mitochondria will be still intact. So this experiment can be translated to the meaning that the tiger milk mushroom ethanolic extract can keep the membrane intact and maintain the status of the healthiness of the mitochondrial membrane. So that's why we can explain that before the apoptosis happened, it had to pass through the process of mitochondrial pathway. And this is again, we can underline the significance of tiger milk mushroom ethanolic extract. Next experiment, we quantify the reactive oxygen species, or ROS, in the cells. So whenever the high level of reactive oxygen species, we can see the high intensity of green color, which is fluorescent color in the cell, which we call intracellular reactive oxygen species. So when we treated the cell with 5 millimolar of glutamate, we can see the high level of intracellular reactive oxygen species. And then if we treated the cells as well as glutamate with ethanolic extract of tiger milk mushroom, you can see here the intracellular reactive oxygen species dropped significantly, about 50%. And this is equal to natural cell in the culture. So this saying that reactive oxygen species happen because of the glutamate accumulation. The tiger milk mushroom ethanolic extract prevent the accumulation of reactive oxygen species, so further prevent the cell death from apoptosis. Cold and hot water extracts of the tiger milk mushroom had no protective effect against glutamate-induced oxidative stress. And the last experiment of the cellular model we study using molecular method, which is real-time or qPCR, to quantify the genes response or feedback to glutamate and also to the tiger milk mushroom treatment. For example, graph A, we check the catalyst messenger mRNA expression. So we found that for glutamate treatment, the mRNA level of catalyst dropped significantly, and then we can restore the gene expression, even a presence of glutamate. But if we have the tiger milk mushroom ethanolic extract, we can restore the gene expression of catalyst significantly, as well as other genes, including SOD1 mRNA, SOD2 mRNA, and glutathione peroxidase mRNA as well. So we can conclude that glutamate leading to the toxicity of the cells and leading to cell death, but the tiger milk mushroom ethanolic extract prevent the glutamate cell death from apoptosis. And then this neuroprotective mechanism involved in the regulation via the antioxidant genes. So the last part of my talk, I will go quickly for the study in the C. ligands, which is in vivo model. So as I mentioned that the C. ligand eat bacteria, OP50 strands. So we have to make sure, number one, that the extract from tiger mushroom do not kill bacteria. So we do susceptibility assay. We make sure that the bacteria are not killed by the tiger mushroom extract. Because if the tiger mushroom cause the bacterial death, then the C. ligands will not have food to eat. So this is very, very important experiment. But anyway, somehow in the past, for this experiment, if you can see that the tiger mushroom kill bacteria, you may find the antibiotics, something like that. So this is the first experiment that we have to do. Survival assay, using cell line, we use glutamate to kill the cell because glutamate causes oxidative stress. In C. ligands, we have to use another compound. They don't use glutamate, and mechanism of glutamate in C. ligands is not so clear. But we use juglone. Juglone is a chemical compound induce oxidative stress in C. ligands. So we have to grow C. ligands L1 larva stage number 1 and synchronize them. And we treated the worm with extracts, hot water, cold water, and ethanolic extract of tiger mushroom, and then treated with juglone. Juglone will increase the intracellular reactive oxygen species, and we count live and dead worms. So I want to show you this picture with juglone 80 micromolar. The number of living worms only 20%. DMSO also toxic to the worms if you use in a high level. But for ethanolic extract, 50, 100, and 200 microgram per mil, and also the positive control, EGCG is the positive control, work to have the number of living worms. Even in the presence of juglone, 80 micromolar, the ethanolic extract can rescue the number of genes significantly, even better than the positive control. So this is very significant experiment. And another graph, right hand, we found that the tiger mushroom hot water extract also work very well. Actually, if you remember, the tiger mushroom hot extract, cold extract did not work well in cell culture model, but it worked very good in C. ligands model. Next, we measure the reactive oxygen species as well. We can stain and see the green color inside the C. elegans. And we found that the intracellular reactive oxygen species, even in untreated controls, very high. But when we treated the C. elegans with tiger mushroom, ethanolic extracts, the reactive oxygen species dropped significantly. And we also found not only ethanolic extract, but cold and hot water extract as well in C. ligands model, that the reactive oxygen species dropped significantly if we treated the C. ligands with all kinds of tiger mushroom in this study. We also check the marker we call stress response protein or heat shock protein. And as you can see, we synchronize the worm treated with the extract as juglone, and then we check the worms on glass slide. As you can see here, juglone induce the expression of heat shock protein, which is the marker for oxidative stress. And then even in the presence of chulalongkorn, but the presence of extract from tigermeal mushroom, ethanolic extract can significantly decrease the heat shock protein oxidative stress marker. And we found that not only tigermeal mushroom ethanolic extract, but also hot water extract, as well as cold water extracts. We check the expression of other markers like glutathione S transferase as well, which is the oxidative stress marker. This is the second marker that we tested. As the presence of chulalongkorn, we actually will increase the expression of GST4, and then the presence of extract, ethanolic extract, will decrease the gene expression of GST4 significantly. And then the next marker we call SOD3, superoxide dismutase enzyme expression. We found that SOD will drop significantly in the cell culture here, but when we treated them with the ethanolic extract and also the positive control, we can increase the expression of SOD3. The SOD gene expression is good because we know that this is antioxidant gene. So you can conclude that antioxidant gene will be increased in the presence of tigermeal mushroom ethanolic extract and hot water and cold water tigermeal mushroom extracts as well. And also we check a transcription factor, DAF16, FOXO pathway also. This gene actually orthodox of mammalian FOXO transcription factor. Actually, when we check them, we have to use different strain of the C ligands, and the expression of this gene alone may not be the answer because the expression doesn't tell that it will work well. So we have to check the translocation from the cytoplasm to the nucleus. So we quantify the expression of this transcription factor, compare between cytoplasm fraction and nuclear fraction or nuclear portion. So I will tell you quickly about this. So based on this study, we have the special strains of C ligands, and we can quantify whether or not the DAF16 is located in cytoplasm or in nuclear or in the intermediate parts. So based on this graph, we found a higher number of nuclear localization when we use the tigermeal mushroom ethanolic extract. As you can see that in the intermediate part, white color, it's not a lot, and also the cytoplasmic part, it's low in the presence of, it's lower in the presence of tigermeal mushroom ethanolic extract, and this is also in line with other extract like cold and hot water extract as well. And we also check the lifespan of the C ligand also, if they can live longer. So based on this study, we study in ethanolic extract, cold and hot water treatment. So we have to count the number of the C ligands. For example, in the first day, we have 100 worms, and we count until the last worm die. So basically, they can live about 30 days. And if we can find longer time that they live until the last one die, we can say that the extract tigermeal mushroom can increase or can prolong the lifespan of the C ligands. We can found, we found actually significant in some of the extract, but not really clear whether or not they can increase the lifespan of the C ligands now. because we also check with the control EGCG as well. So I go quickly. So next we check the aging biomarker, which called lipofuscin. Lipofuscin, as you can see here, when the silicons get older, this autofluorescence will be increased in terms of its expression. We call lipofuscin autofluorescent pigment. So we culture the silicons and then we treated them with the extract. And then over time, we can see that this autofigment of lipofuscin appear in the high amount. But anyway, when there is a treatment with the tiger meal mushroom, as you can see, graph A, we can reduce the lipofuscin marker expression significantly in ethanolic and hot and cold water extracts as well. So we also check the pharyngeal pumping rate, because the silicons, when they get older, the pumping rate may not be so good. But if they are still healthy, the pumping rate is still good. So something like that. So this is the physiological test that normally used for silicon study. And we found that the mushroom extracts work very well in terms of maintain the activity of pharyngeal pump rate in the silicons. So I will go this very quickly. And last, we check the toxicity of the silicon, that we can check the body length. If this extract or natural product cause toxicity, body length will be shorter. And we found that no toxicity from tiger meal mushroom to the silicon. So it's quite safe. And the blood size number of eggs, whether or not the natural product work very well, prolong their life, but cause fertility, something like that. So we check blood size assay, meaning that the number of eggs of silicon, and the results show the normal stage of reproductive activity. We call blood size of silicon. So I would like to conclude that based on 2 model study, cell lines and in vivo silicons, we found pretty good activity, neuroprotection, and also anti-aging of the tiger meal mushroom for in vitro models. The ethanolic extract work best, but for in vivo model, on 3 extracts work fine. So thank you very much for your kind attention. I will take the questions that you may have. Thank you.
Okay. Thank
Okay.
you, Dr. Tewin, for your presentation, for the very established work that you have done in your lab. So I think there's many. I think maybe you have maybe more than 1 PhD student working on that project. Because there's many type of tests.
Yeah.
Essay has been done, right? So
Yeah,
I'm sure
that's
there's
right.
more than 1 student working on it. So okay, while waiting for the question, I have some question for you. Okay, as we know that tiger mushroom is always for the patient, right? However,
Yeah.
in your study, you are working more on the neurodegeneration type of disease. So, can you elaborate how or mechanism maybe between the asthmatic patient and how is the neurodegeneration degenerative can be associated with this asthmatic study?
So can you repeat your question again, please?
My question is, how this tiger mushroom, okay, in your study can be associated with that to
Protection.
cure on the other disease for example, asthmatic. As we know, tiger mushroom, always people talk about to cure the asthmatic disease.
I think the basic mechanism based on this study is still rely on the mechanism that generated by the reactive oxygen species. So I think based on this proposed model, the tiger mushroom may reduce the level of oxidative stress in the cells. Since oxidative stress actually associated with many pathological events in the body, so one way, if we can decrease the oxidative stress, so we can maintain the healthy status of the cells, particularly the mitochondria, for example. If we can maintain the status of healthy mitochondria and other organisms, it can actually maintain the health status of the cells and also the organelles as well. Because actually, I one of the mechanisms that I want to study related to neurological disease and other disease that generated or affected by oxidative stress is the mechanism we call autophagy. That's maybe another pathway that we have to study. Autophagy is the self-killing some organelle or something like that. So if we get rid of the waste product in the cell, and then we can maintain the big portion of the cells, we can still survive without killing the cells. So that could be my answer and explanation for feedback to your question. Yeah.
Okay. Okay. Thank you for the answer. Okay. So the next question related to that: do you think that this tiger mushroom can also be used for the neuroprotective, or for the to protect the cell from the cancer or something? It can be as a protection for the cancer.
For the cancer, right? For which type of cancer? Brain cancer or any kind of cancer?
Any type of cancer, maybe brain cancer will be interesting.
There's some well-performed study in the previous year or previous publication. One of them that I want to take as the example, the tiger mushroom have the ability to reduce the inflammation. So that's very important questions. If those type of cancer happen because of the inflammation, inflammation, when it happens, there's a lot of environment leading to cancer. So if the tiger mushroom can suppress the inflammation, I think there's a very high ability of tiger mushroom to also suppress the cancer. So that could be a good explanation as well. So I'm not sure actually how many papers in the previous period that telling us about tiger mushroom and cancer, but for sure that's one publication really clear that tiger mushroom block inflammation. Yeah.
Oh, that's really interesting. I think we can expand on that research on this kind of work for sure. Okay. There is a question here. Okay. Thank you, Dr. Tewin, for your great presentation. I just wondering, is there any targeted metabolite in tiger mushroom you can speculate responsible for these neurodegenerative activities?
The metabolite, right, in tiger mushroom. Yeah, I actually look at some part of, for example, the fatty acid. Actually, if we found any fatty acid or oleic acid, for example, those type of acids may be responsible for the brain cells target, actually, because in the brain, it composed of the high level of lipids. as the brain have lipid 70% and protein only 30%. So any lipid that can work well in the brain and also function in the brain to control the brain function or cellular function. I think something like oleic acid or other fatty acid could work in the brain and also work as neuroprotectants in the brain. That's what I think. But we still have to find more. Sometimes it could not be a single metabolite. It could be a profile metabolites that work together, synergy in a synergistic fashion. That's my answer. Yeah.
Okay, thank you for the answer. Okay, any other question is, okay, there is one here. Is there any promising bioactive agents in this mushroom that has potential medicinal properties for asthma?
For asthma, I found in the previous publication there's a study, but not sure what they found. I mean, in terms of metabolites. But as I mentioned, if the asthma happen, I'm not sure if one cause of asthma it is inflammation. So if the asthma generated based on the inflammation activity, I think that could be metabolite in the tiger meal mushroom to work against the asthma. But again, in many study in the previous publication, when we study tiger meal mushroom, we did not yet have a lot of fractions to study. So mainly the study based on the big portion of extracts, not narrowed down properly to single metabolites. Since I'm also not sure if we narrow down to single metabolites, if it's gonna work well, I don't know, because we used to work using the extract compared to the single phytochemical, and then the single phytochemical did not really exhibit the activity, but the extract is very good, work best in terms of against the disease, something like that. So that's my answer. Yeah.
Yeah, there is a synergistic effects of the metabolites, the group of metabolites that work together.
Yeah.
Yeah, to work together on the mechanism. Yes. But if there is extract, I mean, if you do for the purified the compounds to the single compounds, so
Mhm.
if there is a good activity or this kind of single compound, okay, it's really a big breakthrough.
Yeah, that's right. Yeah, that's very good. Would be a very good in terms of drug discovery, and then we can just focus on that particular compound. So that would be a breakthrough, as you mentioned. Yeah, thank you.
Okay, there is one. This is another question. Thank you, Dr. Tewin, for the great presentation. May I know the challenges in the study using the C. elegans model in your study? Are they easy to be taken care of? Do you measure the numbers of neurons in the C. elegans?
So for the neuron, we did not measure it yet. We just check the total worms survivals. So I think in the future we will do that, and we will find a proper way to stain the silicons brain part, and we plan to do that. So in order to plan to do that properly, so we now look at the the equipment like a good microscope to do so. So we have to increase our ability, research facility first to do that. But in the future, we really want to do that. We want to take on 3 different systems: the brain, the digestive system, and also reproductive system all together. Yeah. Thank you.
Okay, another question from Aken, our PhD student. Just
Yeah.
wondering, flavonoid is also classified as phenolic compound.
Mhm.
Why there were dissimilarity between each of the extract? Usually we assume that higher flavonoid would reflect higher phenolic, right? Usually we
Yeah.
assume, yeah.
Yeah, but in terms of the assay, there's a way, I mean, different method to check specifically flavonoid and also phenolic compounds. So I'm not sure exactly the difference in terms of the method, but there's well-designed experiment that we can differentiate flavonoid and phenolic. And also, as you can see in the result, the one that have highest phenolic is not the one that highest flavonoid. So in the result that I show you, the one that highest phenolic is ethanolic extract, but the one that highest level of flavonoid is the hot or cold water, something like that. So there is different also in terms of the elements of the assays. So that's why we use different method. It could be more than flavonoid, phenolic also, but we did not complete on of them anyway. Yeah, that's good point. I have to look to that as well. Thank you very much for your comment and question.
Okay. Are any person play a role moving towards extraction of specific classes or molecule from the tiger mushroom?
Okay, can you repeat again, please? The voice not so clear. Thank you. Can you hear me?
Okay, sorry.
It seems the signal lost. Yeah. Can you repeat again, please?
I can hear you. Can you hear me, Dr. Tewin? Hello, Dr. Tewin,
Yeah,
can
yeah.
you hear me?
I'm here. Yeah.
Okay,
Yeah.
okay.
Yeah, I can hear you. Can you hear me? Yeah. Okay.
Okay, I can hear you. So I repeat the question. Are there is any particular classes of compound or method used in the tiger mushroom activity? Are you moving towards extraction of specific classes or molecule from the tiger mushroom?
Yeah, yes. We want to do that now, and we actually we want to narrow down and then subtraction as much as possible. And also we are receiving the new extracts and then also the new mushroom extract from Malaysia also. So now in the current state we want to do that, but you know, in the past 1 year the lab not so good. I mean, we have to stop very often. It seems that there's no productivity in terms of our research in the past 1 year due to COVID-19 pandemics. So even want to do that, but I have to tell you that we do that really slow now. Yeah.
Yeah. Yeah, understand. You are we also the same. Okay, the next question, other than H222 HT22 cell, have you tested the extract on the other type of cell model as well, such as SHSY5Y or HEK293 cell in your study?
Yes, those studies still going on. We want to check different types of cell as well. The HT22, as I mentioned, that we tested was glutamate receptor independent. So we want to also check with other cell types like PC12, for example, and also other models of brain cells. But as I mentioned, it's really slow now. But there's one study from Malaysia, actually. They use PC12 and study neural outgrowth. Since PC12 is a very good model to study neural outgrowth, so you can see the branches of the cells when the neural outgrowth induction happen. So that's another way to study. And then in other cells that you want to do, you have to make sure that those cells express or don't express the glutamate receptor, so we can classify properly whether glutamate dependent or glutamate independent receptor model. But anyway, even glutamate dependent or independent model, we have to make sure that the cells respond to glutamate very well. Or in fact, we can use other type of inducer that cause oxidative stress. but we have to make sure that we mimic the brain function. So in order to mimic the brain, so we use glutamate. We know that glutamate is the endogenous, right? Endogenous glutamate happen, and then it may cause the cell death, and then that's why we can explain for neurological disease like Alzheimer's disease and Parkinson disease. So yes, we do. We want to explore other cell models. Thank you for the question.
Okay, next question. What was the concentration of ethanol used in the ethanolic extract?
The ethanol used, I don't remember. I don't remember exactly, but it's little low, actually. But for the extract, we use ethanol. I know that it's low, and then eventually we remove on ethanol, right? We remove on ethanol, and then we dissolve the extract in the MSO, for example. So I think the ethanol use is eventually get it off by the MSO dissolve. Yeah. But if you're concerned about the safety of the ethanol, that's why we always did the experiment together with the cold and hot water extracts. Although the results not so good in the cell model, we show a very good activity in animal model, which is C. elegans model. So we're concerned about the safety of the ethanol, or maybe some people don't want to use the ethanol. So we always do the experiment using other water extracts as well. Yeah.
Okay. Okay, thank you for
Yeah.
your explanation. The next question, may I know, do you measure the protein from the GFP signal? If yes, how?
Okay. So for the GFP that I show you, you can see the fluorescence. There are different strains of silicon. The silicons have different strains that we use because in Germany the lab is well, perform in terms of silicon lab. For example, if I want to measure the SOD expression, meaning that that strain of silicon will have the high expression of SOD. So they have SOD gene linked to the GFP. So they have the high level of, they have the gene linked to GFP. So whenever we use this model, the GFP, which is gene fluorescent protein linked to the gene, will be expressed high. We can quantify the level of expression using fluorescent microscopy, like confocal microscope. So that we did. And also, if we want to quantify another method, we can also use the Western blot. But using Western blot, it may take time and tedious work. But if we just look at the confocal microscope and then quantify the fluorescent intensity, and also very good in terms of silicon, if you quantify, you not just only quantify, but you can see the organelle, the intracellular portion, where exactly those clean fluorescent protein linked to your protein is localized. So molecular imaging is very important in terms of working with silicons, as well as the protein assays like Western blot. So that's my answer.
Okay, there is a question from the live FB, Facebook. From Kalida. Okay, thank you very much for your insightful presentation. My question is, have you considered to study the effect of tiger mushroom extract, especially water extract, which consists beta glucan and on gut-brain axis relationship?
In the gut, right?
Yeah, gut-brain.
Yeah, we want to do that as well. Yeah, so that's very important questions. I think we will continue doing that too in the near future. So yes, we have to do, well-designed study. It may take time, but in the next few years, I think we can do that, because it may tell us something, the result in the future. But again, one of the things that we have to do is to do a very good control in terms of the extract that we have. Sometimes if the extracted for many months, so many years ago. We don't know the integrity of the extracts. So that's why it's something that we have to keep going with the study, but with a good quality control as well to explain what you just asked. Yeah, thank you. Yeah.
Okay. Thank you for the explanation. I'll check if there is any other question. Okay. There is a question here. In your opinion, does compound in tiger mushroom has the potential to be developed into new target drug for human, or does the tiger mushroom has better beneficial as general supplements?
So now I if we cannot isolate the compound responsible, or we cannot find the metabolite, even single metabolite, to explain what happened in the intracellular level, it could be only the supplement. But if we can isolate or see, the how it works in terms of single metabolite or single phytochemical, that could be a drug. So even in Thailand now, when we talk about this, a supplement could be a easier way to apply to people. But as a drug, it may pass through many steps, including clinical trial in the future. Yeah, it could be a long term or a short term based on this capability to do experiment and also to design and also perform the study. For short term, I believe a supplement is feasible, but for long term, a drug is feasible. Yeah, that's my answer. Yeah.
Okay, I think that's all question in the chat box. We just want my last questions on that C. elegans. Okay, you are mentioned about that we need to do the microbial susceptibility test
Yes.
before before we can do on the C. elegans. This is due because of the C. elegans eat the E. coli, right?
Yeah. Yeah.
Is it because so meaning that if you have the plant extract with that can kill the microbe, so it cannot be used for the C. elegans study, right?
Right.
Okay.
And then that's why very important, because in the past, some group of the researcher study using the plant extract like that. If they don't check if the bacteria get killed or not, and then they find the lifespan increase. The lifespan of silicon increase may not because of the food that they have, or because not the plant itself, but lifespan expectancy, long lifespan can happen because of calorie restriction. So that another theory. If we eat less and we control the calorie, we eat less, we can live longer. We call calorie restriction, because if we eat less and we let ourselves hungry, the autophagy will increase. That will maintain a very good status of health. Calorie restriction leading to lifespan increase. And then if you don't take properly, bacteria kill. Celi can live longer. Maybe this is why, to explain that they can live longer, because they don't have food or lack of food, something like that. So this is the very first step. Yeah. And also another group study this. They know that if they found the natural product kill the bacteria, they discover antibiotics, something like that happened already. So one of my plan, instead of using silicon, we publish as antibacteria plants, something like that. Yeah. So this is first step. Yeah.
Okay, thank you for that tips and also the suggestions. So I think we have come to the end of the webinar. So I would like to thank Dr. Tewin for this very interesting subject, and I'm sure it has opened our view and give ideas on how to bring up your research to the next level. So today webinar has about more than 70 participants. Okay. So in our WebEx and also in the FB Live, INBIOSIS UKM. So I would like also to thank all the participants for spending your time with us today. Please stay safe and take good care of yourself. Before we end the session, please switch on your camera so we can have a memorable photography session. Everyone ready with your camera, with your smiling face? Okay, our secretariat, Tan, if you can capture the photo. Where's Ethan? I cannot see you. Okay, just let us know. Okay, I just count, yeah. Ready? 1, 2, 3. Okay. So I think okay.
Yeah. Thank you.
Mel, okay. Okay. So thank you so much to everyone. See you again in our next webinar session. Please stay tuned. Yeah, please take good care of yourself during this pandemic season. Sorry. So please be safe. Okay. Dr. Tewin.
Yeah. Thank you very much, and
Okay.
send to the
We
best
will
wish
end
to all
this.
Malaysians. And thank you very much. Yeah.
Thank you, and please stay tuned for the attendance form that we have for the e-certificate. Okay, if you want to have the e-certificate, please stay. Thank you.
Thank you. Goodbye. Have a nice day. Thank you.
Dr. Stevis, please stay here.
Yeah, I will stay.
Bring
Okay.
you to the next. Yeah, you bring you to the breakout session. Okay.
Okay. You're fine. Oh, Malaysia sih.